All right, so here is our lecture for Monday, and I gotta admit, this was about the best slides to put together this semester because the chapter we're covering on Monday basically goes back through what the chapter on Wednesday talked about it, but asks, are these really scars from macroevolution, and doesn't assume that universal common descent is true, rather goes and tries to offer an interventionist explanation. So it didn't require me creating a lot of new slides, which was very convenient for me. As you listen through this and as you prepare for our class on Monday, I want you to know that we're going to spend the bulk of our time on Monday going through transitional series in the fossil record, those that are trumpeted as some of the best evidence for macroevolution, and talking about them in more detail. So we'll talk about the whale series, we'll talk about the horse series, we'll talk about the mammal-like reptile, so the synapsid series, and we'll talk about a lot more in detail about the early tetrapods with Tiktaalik, Eusthenopteran, Ichthyostega, Acanthostega. So that's what we're going to spend most of our time on Monday doing, but in preparation for the quiz, make sure you know how to answer all of these questions on all of these areas. So how should we examine evidence from the natural world? Very similar slide to what we had for Wednesday. And again, we need to point this out. We have to evaluate each line of evidence as a separate piece of evidence. We cannot assume that our particular view of origins is true. We need to evaluate each line of evidence and figure out all of the possible explanations for these data, and then from that come to an idea of whether the natural world provides more evidence for universal common descent or more evidence for or against universal common descent. And so as we go through here, we're going to ask, are these really these scars, are these really what you would predict from macroevolution? We're also going to ask, would we predict the very same thing from an interventionist view? We'll ask this question, does an interventionist view actually explain the data as well or even better than a methodological naturalism view? And we need to keep in mind that there are many reasons why somebody would believe macroevolution and universal common descent is true. The chapter makes a big point of this. You need to understand that there are many reasons to believe this. Some people believe it because they maybe came to Christ later in life and they were taught this and had never really been forced to think about Scripture being the revealed Word of God, and then they're already committed to this position. Maybe they're not a Christian at all, but they're not necessarily anti-God. They've only learned a universal common descent, a macroevolutionary view of origins, and they don't really believe that you can take any other view seriously. Maybe they're very honest, and they have studied all four of our hypotheses. Universal common descent is true and follows Darwinian principles. Universal common descent is true and does not follow Darwinian mechanisms. Universal common descent is not true, but microevolution and speciation follow Darwinian mechanisms, or macroevolution is not true, and even microevolution and speciation often need non-Darwinian mechanisms. Maybe they've honestly came and tried to study the data and see which of those four hypotheses are true, and they still came out that one of hypothesis 1 or 2 are true. There are many reasons to believe this, so we can't belittle this idea. All right, so let's go through these scars, and again, we're going to ask, are these really scars? And so this whole idea of homology. So homologies are oftentimes presented as strong evidence in support of universal common descent. Now, what's interesting here is you do have to admit that homologies, which we're going to take the structural definition or the structural definition of a homology. So a structure that may be used for different functions but has a similar internal structure, design, and development. While that does makes sense if all of these organisms came from the same ancestor. It makes just as much sense if all of these organisms came from the same designer. You have to remember that there are developmental constraints. And so if all vertebrates were designed a particular way on a particular pattern or under a particular model, that there really isn't, you really wouldn't necessarily expect that every single group of vertebrates is going to have a completely different development and design, but rather has a very consistent design that then is robust enough to modify and take on different functions. And so it can very easily be explained as an interventionist but view. Now, is this a prediction that we would make under an interventionist view? Probably not. At least it's not one that I would necessarily think. Although I think we have just as good of an explanation for its existence as you have from a universal common descent view. Again, that these common structures are not because of common ancestry, but because they're from common design. That the designer used a model that was robust and flexible and could diversify. And then, so why do we have analogies then? So analogies, you've got a structural definition of this, a structure that carries out the same function but with a very different internal structure and development. And so here we've got these organisms that are built with different models. Some of these are vertebrate organisms. Here are some vertebrate organisms, but again built on slightly different models. And here's an invertebrate built with a very, very different developmental model, and yet they were all designed for flight. And so you have these organisms that were built with slightly different design models, and then they were all designed to fly. And so they have to develop differently because they have a different model, a different design, and you're going to end up getting different structures carrying out the same function. Really, this isn't a problem. The problem here is, why would entire sets of organisms be designed with a particular model? That's maybe a more interesting question, and one we'll deal with a little bit later. So are homologies, scars of macroevolution? Well, really, only if you are committed to a methodological naturalistic view of origins, we can explain them just as easily from an interventionist view. What about these biochemical, these biochemical homologies? So here, a reference to cytochrome C and the number of amino acid differences, and we see that there's basically the same protein appears in almost all living forms, and has a pretty decent amount of even sequence homology among the different groups of life when you compare the amino acid sequence. And so an interesting point to make is, if these organisms are designed with a particular model that then diversifies, and there is structural similarities among these forms, you have to remember that structure, anatomy, is not independent of biochemistry. In fact, anatomy and biochemistry are highly correlated. So if you have anatomical similarities, you would expect that you are going to have biochemical similarities. And then further, if biochemical similarities are not independent of genetic similarities, so if you have biochemical similarities, you will have genetic similarities as well. But something that's really interesting is when you look at this, and here's a chart modeling some of these relationships among these various groups. So you see here, as we get groups that are isolated from each other. So here we have 15 differences between the lamprey version and the horse version. But you could see that this lamprey actually has pretty consistent differences when compared to these other groups. And I'll show you another graph where you'll see this even more so. So here are these bacteria. So if you trace them to all sorts of different, any non-bacterial organism, whether it be a horse, a pigeon, a tuna, a silkworm, very different groups of life, the bacteria basically contain the same number of differences from all these non-bacterial forms. And we have the same relationship. If you compare wheat to horse or pigeon or tuna or silkworm, that wheat is equally different from all these different groups. And so this is definitely not something that you would predict from a universal common descent view, and or maybe it would be, because at least these groups are going to be equally distant from wheat, silkworm. All of these are animals. They're going to be potentially equally distant from wheat. This is very interesting. And then, silkworm, and you compare that to all these different vertebrate groups. And again, we have a very consistent difference. And what gets really, really interesting is this chart here. So here you have two different versions of a similar protein, relaxin, which in mammals functions to allow the cervix and the birth canal to get ready to give live birth. In other animals is not used in the same way, but you find it in other groups of organisms, especially in cartilaginous fishes. And so you can see the amount of variation here, the amount of different versions of this. And so mammals have both relaxin A and relaxin B, and you can see it shaded in the same way. And cartilaginous fishes have both forms of these proteins, and this is representative of the amount of variation. And so what you find is, thought I had that in the chapter. They have a table that shows some of these differences, but the amount of difference between the human version of this protein and the mouse version of the protein are just as distant as the human version of the protein is from the shark version of the protein. And this is certainly not something that you would predict from a perspective of universal common descent, but is something that you would regularly predict from an interventionist view, that these organisms were created in such a way that a number of the design elements were recycled or reused in different groups. And oftentimes you're going to find examples where things just don't make sense, right? The mouse version should be far more similar to the human version of this protein than the shark version is to the human protein. And yet, if universal common descent is true, and yet they aren't, indicating that both of these groups of organisms were designed with this protein in place, which has diversified through time. So we have a new question, even just dealing with phylogenetic trees, period. Do we have a reason to doubt phylogenetic trees? So something you need to know is, in order to build a phylogenetic tree, you have to use an outgroup. You have to use a group that you don't consider to be part of your phylogenetic tree because of ancestry in order to create this rooted tree. Otherwise, your tree looks like this. And so in order to even build a phylogenetic tree, you have to assume that all of these groups in your phylogeny share ancestry, and that they all share ancestry with this outgroup, but that outgroup is not part of this group. So you can't even build a phylogenetic tree unless you assume the universal common descent of at least all of the organisms in your phylogeny along with that outgroup. Which this is interesting. So a phylogenetic tree can only ask the question, is this tree true? It could never ask the question of should we even build this tree? It can only ask the question, does this tree match the data the best? It can't even ask it like, are these organisms even share an ancestry? Because you have to assume they do in order to build the tree. And here you can see in A, B, and C, 4 different trees that are generated by changing your particular outgroup. So which group you decide is most closely related to all of the members of your group is going to have a huge bearing on what your tree actually looks like. And then in D, we don't provide an outgroup, and what we find is 1 and 2 groups similar, 3 and 4 groups similar by either morphological or biochemical or genetic characters, or maybe all 3. But since we didn't provide an outgroup, we don't actually get a rooted tree. And instead, this is functioning just to show similarities and not evolutionary history. So do we have a reason to doubt phylogenetic trees? We do, because in order to build them, you have to actually assume that the organisms share ancestry, which is interesting. So some more of the scars of macroevolution. One of these things we talked about development for Wednesday and the shared developmental forms. Again, we've already addressed the idea that it seems to be that vertebrates were designed with a particular pattern that then could diversify through time. And so we shouldn't expect to see, we shouldn't necessarily be surprised to see some similarities. But one thing you have to know is pointed out in this chapter, and I've mentioned it before, a number of these drawings done by Haeckel and his students overemphasized similarities, and there are some very, very significant differences. Anyways, and then also a number of developmental similarities can be explained by what is functionally required, especially when you talk about the primitive kidney that you find in humans and other vertebrates that develops early on and clears out metabolic waste from the early embryo that then gets replaced by the really derived sophisticated kidney. Well, the early embryo needs a simple kidney, and so we have functional and developmental constraints in place that require that. Here we talked about this as being evidence that birds evolved from reptiles because they still have the developmental capability to develop the reptile skeleton. Now this one's interesting, and the book addresses this. If birds have not used this developmental pattern for over 100 million years, why do they still, how did that genetic ability to make it persist, and how wasn't it not destroyed by random mutations? Which is a very interesting question and a potentially very difficult issue to deal with. And so they argue that what this looks like is again more evidence of this very consistent design pattern put in all vertebrates that was robust and flexible enough to generate the different designs necessary for our different types of organisms. Oh, the vestigial traits, and I mentioned this last time in class. The authors of our textbook, I'm pretty sure it's Brand, calls these vestigialism of the gaps arguments. That basically what we mean by this is that we don't know what the function is yet. So the nictitating membrane does not function as a third eyelid in humans, but rather functions some way with the process of producing tears and keeping the eye moist. We don't know exactly what it does yet. These segmented muscles, rather than just being unnecessary remnants of our evolutionary past, work a great deal towards keeping us upright and in our upright posture. The appendix is a place where healthy gut bacteria reside and in some way carry out their normal function, keeping our normal gut bacteria healthy. Our caudal vertebrae, a necessary place for muscle attachment to help keep us upright and to cushion us when we sit. These muscles of the nose and ears, they don't seem to be used in most people, but maybe were earlier in human history. Here, the wisdom teeth, third set of molars, again, we talked about this, that maybe these are truly vestigial and we used to be larger than we are now. Hind limb bones in whales, and many of these function in reproduction as a place for attachment of reproductive structures. And so vestigial structures are tough. Again, if you look at a list of vestigial structures from 100 years ago, it's much, much longer than our list is today. Some of the scars. So inefficient designs. This one is an interesting argument. Oftentimes you get arguments like, hey, I could design something way more efficient than this, like the panda's thumb. It's just so inefficient. What's interesting, because the pandas don't seem to mind it at all. And again, it assumes that the designer could, or of course he could design the thumb however he wanted, but that he would design things in a very particular way. Again, this is a bear and was built with a bear model, and therefore, if it's going to develop a thumb, has to develop a thumb from the bear blueprint. And so it seems to be very consistent with how God designs other organisms. And so, is this something that we would predict from an interventionist view? I certainly would. That if you wanted to build a bear with an opposable thumb, I would predict that it would be built with a normal bear design and not with, just adding a digit and having an opposable thumb like a primate's thumb. That's exactly what I would predict, and I think we have just as good an answer for it as you have from universal common descent. This one, why does light travel through several cell layers in order to get absorbed? This has been, dealt a very significant blow in that the cells that line the retina are incredibly efficient at transmitting light through those cells to where that light gets absorbed. Really, really efficient at doing that. And this allows the blood supply for these cells to be back behind the retina and doesn't force the light to travel through the blood, which is very opaque and does not move through very efficiently. And so it actually is an incredible design to make light absorption very efficient. Right as the recurrent laryngeal nerve go into the chest and around the aorta and then back to the larynx. This is especially dramatic in something like a giraffe. But again, it assumes that giraffes always had long necks like that, which I don't think is necessary to assume. And again, from an interventionist perspective, I would predict that there is a very common design element in particular groups that then is modified during development for the different needs of that organism. So I think we have just as good an explanation from this as an interventionist view as a universal common descent view. And there, the hierarchical organization of life. This is oftentimes trumpeted as an enormous example and a powerful example and evidence in support of universal common descent and macroevolution. But what you find is this is the way ecosystems are structured. Ecosystems are hierarchical. And so if you're going to design a group of organisms being vertebrates that are oftentimes that are going to have the flexibility to be at various places within the ecosystem at various trophic levels, this is exactly what I would predict, that if ecosystems are going to be hierarchical and every ecosystem has very similar niches to others, and you need organisms that are flexible enough to fill those different niches, but we should be able to, if an organism is filling, let's say, is in the second trophic level and feeding on organisms in the first trophic level and multiple different types in one ecosystem, in another ecosystem, an organism that's at second trophic level feeding on multiple different types of organisms from the first trophic level, I would expect that those two organisms at trophic level 2 in the different ecosystems are going to share a great deal of similarities and probably could be grouped together based on common features, and you're going to end up getting this hierarchical nature of life. And the argument that a designer wouldn't design this way is always going to be a hard argument to make, because we have no idea all of the ways in which God could have designed creation. All we have is a evidence of how God did design it, but how that design has been impacted by the fall and by sin and by degradation. So we don't even have a clear picture of how God actually did design it without those imperfections. So, biogeography, this is a really difficult one. This is one that the textbook saves for a later chapter, and so we'll save most of our discussion for this. But biogeography, there are some, good. Most of biogeography is explained the same way, whether you're an interventionist or you are a methodological naturalist. That most of it is explained by animals moving from one place to another, as the different finch species on the Galapagos Islands. Nearly every interventionist would say, yeah, they came from the mainland, and then as they spread among that archipelago, they diversified into the different species that you have. It's not really something you wouldn't predict from an interventionist view, but major groups of, like, why is virtually every mammal in Australia marsupial, and why do Australia and South America, the furthest places from where the ark would have been, have the most unique forms of life. And so some of these are interesting and difficult questions to answer, but we'll save those for a later understanding. The fossil record. So again, we talked about the fossil record providing some evidence in favor of macroevolution, especially with this increase in complexity and increase in variety as you move up the fossil record. But again, that is not necessarily something I wouldn't predict from an interventionist view as well. Now, one thing you would probably predict is if modern mammals that we have living now were present at the very beginning of creation, we should at least expect to find some early in the fossil record. And at least I would, but it requires an understanding of even how things get fossilized and how that process works. And again, we'll save a lot of this discussion for another time. But one thing you certainly would not expect from a macroevolutionary view is that you have a number of varieties showing up all at once with no transitional forms, no intermediate forms. And so plants are a great example of this. We've got four major groups of plants. You have your non-vascular seedless plants like mosses. You have your vascular seedless plants like ferns. You have your gymnosperms and you have your angiosperms. Four major groups of plants. And I listed them in order of increased complexity, and they actually show up in that order in the fossil record. The bryophytes, the non-vascular seedless plants, show up first. The vascular seedless plants like ferns show up next. The gymnosperms show up next. The angiosperms show up last. And so that's exactly what you would predict from a methodological naturalism, from a macroevolutionary view of origins. However, what you wouldn't expect is that when they show up, to show up with an enormous variety and no intermediates between, say, a vascular or non-vascular seedless plant and a vascular seedless plant, with no intermediate forms to connect the two. That is certainly not something you would expect, but something we find throughout the plant fossil record and something we find throughout the animal fossil record as well, from the very beginning in the Cambrian explosion, where we find basically all of our modern animal phyla represented with no intermediate forms. And so, yes, the fossil record does show evidence in support of macroevolution, but it also shows evidence in support of interventionism. And then this thing we're going to talk a lot more about on Monday, so I'll save most of our discussion for there. But are these really scars of macroevolution, these transitional forms? So we've got our therapsids, our mammal-like reptiles with seeming movement of bones from the lower jaw into the middle ear, replacing, or not replacing, but adding to the one middle ear bone that reptiles have, getting to the three middle ear bones that mammals have, and reducing the number of bones in the lower jaw. And we actually find some forms that look like they are in the process of making the shift. It's very interesting and very convincing that we could generate mammals from these therapsid mammal-like reptiles. But again, we'll save that discussion for Monday. Modern whales, we have generally a very consistent and predictable sequence, moving from a terrestrial organism to a semi-terrestrial or semi-aquatic form to a fully aquatic form that still has some terrestrial features to fully aquatic form with absolutely no terrestrial features. The organisms show up in the right order in the fossil record. They show up without an enormous diversity. They show up with the diversity increasing as you move through, extremely convincing until you note that we actually have some relatively modern whale forms that now we've found in deposits nearly as old as this terrestrial form. So we have to talk a little bit about this whale. Actually, we have to talk a lot about this whale series and talk about, how convincing is the evidence. So one thing we mentioned in a previous class is that an interventionist view tends to hold that the original forms are more complex, and then as you develop and as you speciate and the organisms specialize, you're going to lose a lot of that variation, some of that complexity. And what's interesting is as you look through vertebrates and as you move to what are considered the more primitive vertebrates from a macroevolutionary view to the more derived vertebrates, you see a reduction in the skeleton, the number of bones making up the skeleton. Is that a reduction in complexity of the skeleton? Maybe, although with fewer bones, you increase the amount of flexibility, you increase the amount of space for muscles, and but there's not really a good explanation for this from really either view. I mean, this is just kind of an interesting conundrum. So again, as you prepare for Monday, keep in mind that we're going to focus in on these transitional series, and we're going to really talk a lot about those. But be prepared to answer questions from any section in these slides.